Welcome student. Now in this class we are going to study methods of plant breeding in self pollinated crops. The main objective of plant breeding is to produce the new varieties with superior in all aspect than the existing one. The principal methods of breeding pollinated crops are: first one, introduction and acclimatization, and then selection, hybridization or the method, and then mutation breedings and breeding. First we are going to see the introduction and acclimatization. In introduction, so here two types of introductions are here: a primary introduction and secondary introduction. Introduction may be involve new varieties of crop already grown in the area, while relatives of the crop species or totally new crop species for that area. Plant introduction may within the country, between the countries, or confirmed between the states or within the state. The plant may be introduced from the country of another continent. Example: introduction of redley wheat varieties from Australia. Introduction may be classified into two categories: primary and secondary. So first we'll see the primary introduction. When the introduced varieties is well suited to the new environment and is directly released for the commercial cultivation without any change the original genotype, known as a primary introduction. Example: introduction of semi drop wheat varieties Sonara, Lerma, Rojo, and semi drop rice were TN1, IR8, IR28, and IR36. Then secondary introduction. The introduced varieties is subjected to selection to isolate superior variety, or may be hybridized with the local variety to transfer one or few desirable character to the local variety, known as the secondary introduction. Procedure of plant introduction. Plant introduction is one of the very old and effective method of plant breeding. It consists of following step: procurement of germplasm and quarantine, cataloging, evolution, multiplication and distribution, accumulation. So we'll see the accumulation, the process that leads to the adoption of a variety to a new environment, is known as acclimatization. Generally, the introduced varieties perform poorly because they are often not adapted to the new environment. Sometimes the performance of that variety improve in the new environment by growing it for number of generation. Acclimatization is brought about by a faster growing it, or for a number of generation. Accumulation is brought about by a faster multiplication of those genotypes that are better adapted to new environment. The population having more variability is easily accumulated. That is cross pollinated crops are easily accumulated than self pollinated crops. Merits and demerits for plant introduction. First, we'll see the merits. It provides superior variety directly after selection. It provides the variability from genetic erosion by collecting germplasm. It is very quick and economical method of crop improvement. It provides protection to the crop by introducing into new disease free areas, that is coffee and rubber. Demerits: introduction of noxious weeds, introduction of diseases, introduction of pest, ornamental turns weeds, threat to ecological balance. So in next part we are going to study, that is the selection of plant. Selection has a two basic characters or limitation. Selection is effective for heritable differences, selection does not create variation, it only utilizes the variation already present in the population. Thus, the two basic requirements of selection are variation must be present in the population and variation must be heritable. So here there are two types of methods for selection: pure line selection and mass selection. So here pure line selection, uh, the pure line selection concept is um, discovered by Johnson's pure line theory in 1903. So pure line means it is the progeny of single self-fertilized homozygous plant. Pure line selection. In a pure line selection, large number of plants are selected from self-pollinated crop and is harvested individually. Individual plant progenies from them and evaluated separately, and the best one is released as a pure line variety. Therefore, it is also known as individual plant selection. Characteristics of pure line: all plants within a pure line has the same genotype as the plants from which the pure lines are seride. The phenotype differences variations within a pure line is environmental and therefore non-heritable. The pure line becomes genetically variable with time due to mechanical mixture, mutation, etc. Uses of pure line: superior line is used as a variety. It is used as a parent in development of new variety by hybridization. Pure lines are used for studying mutation and other biological investigations such as medicine, immunology, physiology, and biochemistry. Procedure for pure line selection: the pure line selection has three steps. First step is selection of individual plant from local variety or from mixed population. uh, Visual evolution of individual plant progenies, and third one is yield trials. So here first year what we have to do, the select a large number of plants from desi or local variety or some other mixed population, and their seeds are harvested separately. In case of individual plants can't be identified, individual heads by may be selected on the basis of easily observable characters such as flowering, maturity, duration, disease resistance, presence of arms, plant height, etc. It is advisable to select plants for easily observable characteristics. And what we have to do in second year: selected individual plants progenies are grown with proper spacing week along with standard variety row. Progenies are evolved visually and to poor, weak, and defective. Segregating progenies are rejected on the basis of visual character. The number of progenies selected should be less to facilitate replicated yield trials. If necessary, this process may be repeated for one or more years. The next third year, what we have to do: grown the selected progenies in a replicated trials for critical evolution. The base variety is used as a check for comparison and plant after every 20 to 25 progenies. If sufficient seeds are available, preliminary yield trial may be conducted. Selection is made for easily observable. Preliminary yields trial may be conducted. Selection is made for easily observable characters including disease resistance, and number of progenies are reduced. 4th to 7th year, replicated main yield trials are conducted using best variety as a check. Quality test is also conducted and used as a basis of selection. Each progeny is an experimental strain as it is pure line. The promising strains are evaluated at several locations along with other strains in coordinated yield trials, the most promising strains are identified. 8 year, the best progeny is released as a new variety and its seeds is multiplied and distribution to farmers. Merits of um, pure line selection. Pure line selection achieves maximum possible improvements over the original variety, being extremely uniform, more liked by farmers and consumers than those developed by other methods like mass selection. It is easier than hybridization, requires less skills, used for developing inbreed lines and pure lines. Due to extreme uniformly, it is easily identified in seed certification. Demerits of pure line selection: it is not practiced in cross pollinated crops because it is expensive and laborious. The variety developed can't be easily maintained by the farmers. The varieties developed by pure line selection don't have wide adaptability and stability in production. The upper limit on the improvement is created by the genetic variation present in the original population. It requires more time and laborious than mass selection. The breeders has to devote more time to pure line selection than the mass selection. Application of pure line selection: it is used for improvement of local varieties have considerable genetic varieties, example wheat. It is practiced in introduced material to develop suitable varieties, that is the shining example is shining mung. um, it is used for improvement of old pure line varieties. Example is Safa. It provides an opportunity for selection of new characteristics such as a disease resistant grain type, plant type, etc. It provides an opportunity for selection in the segregating generation from crosses. Achievements: a large number of improved varieties have been developed in self-pollinated crop like wheat, barley, rice, pulses and oilseeds, cotton and many vegetable, etc. Many wheat varieties developed include NP4, NP6, NP12, NP28, Mungwar, T1, B1, tobacco, uh, cha- Chatham, special line, etc. So this is about the pure line selection. So we studied the introduction of the pure line selection, introduction of pure line selection, and then characters of pure line selection and uses of pure line selection we have seen, and then next procedure of pure line selection, and then next merits, demerits, application and achievements of pure line selection. Thank you.